greatest tragedy in the history of medicine. I just, once people wake up to realize what I am saying, and I tell you every this, I show you all the evidence, I did the experiments, I show the efficacy, can't be true, can't be true. That's why I don't understand how some oncologists or even physicians would say that glucose has nothing to do with the growth of the tumor. It absolutely does. It's in the literature. Most important drug that we know of to manage cancer is not being used because they didn't do it right. Interference with what I'm saying has nothing to do with science. It has to do with policy and acceptance of the scientific facts, not the science itself. The science, to my mind, is quite solid.
Dr. Thomas Seyfried is a professor of biology, genetics, and biochemistry, and his remarkable work and research has changed our understanding of cancer. Dr. Seyfried, thank you very much for being with us today. And my first question for you, Professor, is cancer is the second leading cause of death worldwide, with millions of documented cases each year and millions of deaths each year. In fact, in your book, you suspect that cancer will be the leading cause of death in industrial society, overtaking cardiovascular disease as the leading cause of death. In your opinion, what is the reason behind this surge of cancer?
Thank you very much for having me today. And it's a pleasure for me to answer your questions. Yes, and in some countries already, in China, I believe cancer has surpassed cardiovascular disease as the number one killer. And I think you're correct. The trajectory says that it will probably overtake heart disease in many industrialized countries, unless something is done. In the United States, the new information just was released by the American Cancer Society, telling us that over 612,000 Americans will die from cancer in 2025, which comes out to be 1,700 persons a day. And if you go by the hour, that means about 70 people per hour will be dying from cancer in the United States. So, and it's getting worse. It's not getting better. So what is responsible for this incredible increase in cancer? As you said, well, it's never one thing. When you have a disaster like this or an epidemic of whatever, there's never just one thing that contributes to the crisis or to the disaster. Let's put it that way. And this is a medical disaster of monumental proportions. We're seeing younger and younger people getting cancer, so clearly we have an issue with that. But we have several biological explanations, and we have policy knowledge explanations. And let's go through the explanations for why we have a continued increase in cancer. The obesity epidemic is now replacing smoking as a major risk factor for cancer. Obesity is linked to type 2 diabetes and systemic inflammation in the body. Systemic inflammation damages mitochondria. Mitochondria is the organelle in the cell that's responsible for efficient energy production. Disruption of that organelle's ability to produce energy efficiently is the origin of cancer. So when that organelle becomes corrupted in a variety of different ways, from inflammation, from radiation, from chemical carcinogens, from oncogenic viruses, intermittent hypoxia, as I said, these kinds of things, and age by itself, aging. Any one of these provocative agents can interrupt the function of energy efficiency, leading to compensatory glucose and glutamine-driven fermentation and dysregulated cell growth. So that's the origin of cancer. Now, why is it that the numbers keep increasing and the deaths keep increasing? Because the World Cancer Institutes, from the National Cancer Institute in the United States to the NHS in England to all the major academic and research centers, they all are under the belief that cancer is a genetic disease. And when you take the world power of of treatment and focus on something other than what the disease is, you have no successful management. So, so when people say cancer is a genetic disease, they're focusing on things that are not central to the problem. Most of this stuff is secondary downstream effects. The mutations that we see in cancer, the development of precision medicines to try to target these mutations and the consequences of the mutations, all of this is downstream epiphenomenon. If it's not central to the problem, then the problem persists. The problem does not go away. We know that cancer is driven by a fermentation metabolism, that's energy without oxygen, and the fuels for that are the sugar, glucose, and the amino acid glutamine. And we know that the cancer cells cannot burn fatty acids or ketone bodies. So once you realize that all cancers that we have looked at have a problem in generating energy through oxygen and are all dependent to some degree or another on fermentation of two different fuels and cannot burn fatty acids or ketone bodies, then the appropriate approach to managing the disorder is to simultaneously restrict the two fuels driving fermentation and transition the whole body over to a fuel, ketone bodies and fatty acids, that the cancer cells can't use. And then they will gradually be eliminated slowly. And this will drop the death rate and will make cancer a very manageable disease. But the reason why that's not being done is because the field thinks cancer is a genetic disease, and they're not focusing on what I say. And then, and why is that? Because ideological dogma. People have been indoctrinated into thinking cancer is a genetic disease, and any form of indoctrination, whether it's political, religious, or scientific, prevents the brain from being rational.
If you want to learn how to implement the ketogenic diet and the Press Pulse protocol into your cancer management, just visit school.com/keto for cancer, the new community by our research analyst Johnny Rockermeyer. Within this new community platform, you can connect with other patients and learn from their experiences. You can also just search through the posts other members have already made. You will have access to our classroom section as well, which provides practical advice and tools for the best results. These sections cover all parts of the Press Pulse protocol that you should implement. Starting with the general scientific concepts by Dr. Thomas Seyfried, we will show you how to start the ketogenic diet, including which foods to choose and which to avoid. Additionally, we prepared a list of doctors specialized in metabolic therapy, as well as details about glutamine inhibition and hyperbaric oxygen protocols. So, take a look around at school.com/keto for cancer.
You have to have rational thinking to break through dogma. That's the way I look at it. And as long as people are dogmatic in thinking cancer is a genetic disease, the numbers of dead people will continue to increase year in and year out. That pretty much summarizes 100 years of scientific research in this area.
You explained it very clearly. Thank you for this detailed answer. And in pathophysiological term, what is the origin of cancer?
The origin of cancer comes from carcinogens. It comes from intermittent hypoxia. It comes from systemic inflammation. It comes from oncogenic viruses. There's a lot of ways to damage oxidative phosphorylation.
Does chronic consumption of highly processed carbohydrate leads to mitochondrial dysfunction?
It leads to systemic inflammation. Inflammatory cytokines can damage mitochondria. Okay, so it's the mechanism by which mitochondria damage.
Can cancer cells survive without glucose and only glutamine?
They can survive, but they can't grow. If I take away the glucose, the glutamine will keep them alive to some extent, but they can't grow because they need the carbons from the glucose molecule to grow. If I have glucose and take away the glutamine, the cancer cell can survive, but it can't grow because it needs the nitrogen from the amino acid to grow, to make new DNA and RNA. It's the two fuels together that are absolutely essential for the continued dysregulated growth.
And there is a lot of confusion about glucose. What does glucose do to the mitochondria?
Glucose fuels mitochondria. You and I are burning glucose right now in our mitochondria, okay? But if we had cancer, glucose would be fueling the cancer as well. So, but the cancer cell needs the glucose. Absolutely, it's a requirement. Our normal cells can switch to fatty acids and ketone bodies, which the cancer cell cannot use. So then you can marginalize the ability of that cell to survive by taking away the glucose and also the glutamine. It has to be done together because some cancers require more glucose than glutamine, other cancers more glutamine than glucose. So if I take away both fuels simultaneously and transition the body over to ketosis, nutritional ketosis, then you can kill the cancer cells. But you have to be careful because glutamine is an essential amino acid required for the health of our gut and our immune system. That's why we developed the press pulse therapeutic strategy, which chronically stresses glucose availability, but intermittently targets glutamine. Because kill cancer cell, keep immune system healthy. Kill cancer cell, keep immune cells and the gut healthy. Kill cancer cells, keep the immune. So on, off, on, off, on, off, on, off. It's a pulsing. If you try to chronically target glutamine, you paralyze the immune system and damage the gut. So you have to know basic physiology of all organ systems, how they work together, and the vulnerability of the tumor cell. And based on that understanding, all major cancers can be managed. Whether or not they can be cured, I don't know, but we can certainly manage them more effectively without toxicity. That's important. Glucose is not a carcinogen. People have to realize that. They misunderstand what glucose is. If you have cancer, then glucose becomes a fuel. But glucose is not participatory in the origin of cancer. What glucose can do is it can contribute to diabetes and obesity, which create inflammation in the bloodstream, which then damages oxidative phosphate. But glucose itself is not a carcinogen. But glucose is, if you have cancer, which is dysregulated cell growth, glucose is the fuel driving dysregulated cell growth. So if you take away glucose, that shuts down a primary fuel for the dysregulated growth, but not the other fuel. You got to target glutamine as well.
And how do we prevent or lower our risk of cancer?
Put it this way, it's very, very difficult to get cancer if the mitochondria in our cells remain healthy and highly functional. And generally, that's prevention. So our Paleolithic ancestors rarely, if ever, had cancer. Rare, it would be very rare. And how do we know this? Because Albert Schweitzer, a humanitarian physician, studied primitive tribes that live according to their traditional ways, where cancer was extremely rare. Cancer exploded in those tribes when the great American diet and lifestyle entered their environment, and then you get super fat, demented cancer, type 2 diabetes. It's clearly related to diet and lifestyle. But people who live according to Paleolithic ways have low blood sugar, a lot of exercise, and they have to be burning ketones. Our Paleolithic ancestors, because they had no highly processed carbohydrates in the environment, they ate mostly meat, which doesn't jack up glucose very high. They had no sweet foods or processed carbohydrates. Maybe fruit during seasonal times when it would be ripe, they might be able to raise some blood sugar. But they had a lot of exercise, and they ate foods that were very low in carbohydrate content, and they would not have cancer because we see that in tribes that live according to traditional ways. So highly processed carbohydrates, lack of exercise, stress, poor sleep, all allow the body to go into a state of dyshomeostasis, leading to mitochondrial damage, which can be leading to not only cancer, but dementia, type 2 diabetes, cardiovascular disease. All these chronic diseases are the result of metabolic dyshomeostasis as the result of an inappropriate diet and lifestyle.
An inappropriate diet is mostly related to highly processed carbohydrate, food that spikes the blood sugar and keeps it up.
Yeah, that's right. That's what we call high glycemic foods. We can eat carbohydrates, not like some plants, vegetables and things. They will have carbs, but they can be complex carbohydrates. It doesn't spike right away. The spike comes from the glycemic index of the food that you eat. So if I spike sugar by eating a highly processed carbohydrates with minimal or zero nutritional value, it causes my insulin to spike. Insulin spikes right after you put and that drives the sugar into the cells, and insulin becomes insensitive if you keep doing this. And then you start to collect all this fat because the sugar is being driven, is forming fat. Fat creates inflammation in excessive amounts, of course. And you put yourself on the path for chronic disease, cancer being the worst one, because that'll kill you if you don't know how to. And so you have an inappropriate way to understand inappropriate response to a disease that can be prevented.
In one of your prior interviews, you've mentioned that your colleague in Istanbul have used the metabolic therapy in combination with the standard therapy, and that led to better outcome and lower dose of chemotherapy. Did I get this correct?
Yes, you're absolutely correct. The idea though is that if the body is in nutritional ketosis, and we developed the glucose ketone index calculator to allow people, whether with or without cancer, to know whether or not they're in nutritional ketosis. Nutritional ketosis can both prevent cancer and help to kill cancer cells along with glutamine targeting. So we found that a glucose ketone index of 2.0 or below puts tremendous metabolic stress on the tumor cells, making them more vulnerable to chemotherapy, which can be delivered in much lower dosages. So collateral damage to normal tissues becomes much, much less because the tumor cells are now super vulnerable to chemo because their fermentation metabolism driven by glucose and glutamine is weakened. So they are on the threshold of dying. So even a lower dose of chemo, even a lower dose of radiation, even a lower dose of immunotherapy, any of these traditional, more traditional or standards of care will work so much better with so much less toxicity if the body is in a state of nutritional ketosis, because that puts tremendous pressure on the tumor cell survival. So you don't need to use so high of a concentration of radiation or chemo to kill the tumor cells. And our friends in Istanbul are experimenting with these conditions right now, and they're the ones who are relaying this information to me.
And could you tell us why is the metabolic therapy so effective?
Because the cancer cells require the two fuels, the fermentation fuels, and they can't grow, and they can't grow without these two fuels. So they can't use anything else. So metabolic therapy restricts those two fuels, killing many, many tumor cells and making other parts of treatments much more effective. So without those fuels, you can't make new lipids, proteins, or nucleic acids. You need building blocks. Number one, you need building blocks, and number two, you need energy. And without energy, nothing can grow, nothing can live. How do we know that? Because we know cyanide is a poison. People who take cyanide are dead instantly because cyanide shuts down all the energy in the cell. Okay, you die. Without energy, you die. Cancer cells require energy, but they use an alternative form. They use substrate level phosphorylation, basically a fermentation metabolism, rather than oxidative phosphorylation. And we have found and published that the glutamine can generate energy in the mitochondria using a substrate level phosphorylation or a fermentation metabolism to replace the lost energy from oxidative phosphorylation. Interestingly enough, if a person has a tumor, this was done by Warburg and others, and us too, and give the animal the cyanide, it kills the mouse or the host, but not the tumor. The tumor can live without oxidative phosphorylation. So that tells us it must ferment. What does it ferment? Glucose and glutamine. How do you know this? Because we tested it. We tested all the different amino acids to see which one is the most effective, and glutamine is the most effective. So you target the glutamine and take away the glucose and transition to ketones. The tumor cells die. They can't live, and then you know how to manage the cancer. It's not that complicated. It was made to be complicated, massively complicated, because people thought cancer is a genetic disease. And there's millions of different kinds of mutations, making a bewilderingly complicated disease. But when you look at it based on the energy and bioenergetics, it's a very simple disease. is driven by fermentation. And then you say, well, if that's known, why don't we target fermentation metabolism in cancer patients and manage the disease? And the answer is because the field thinks cancer is a genetic disease. They're brainwashed into thinking it's something other than what it is. And therefore, we get 1,700 people a day dying needlessly from this, and around the world as well. So, I mean, it's the greatest tragedy in the history of medicine. I just, once people wake up to realize what I am saying, people are going to just be so mortified by the fact that we had a process, we have a way right now to drop this death rate and keep people alive longer, and we are not doing that.
Thank you very much. Can you stop tumor growth just by lowering the blood sugar?
It doesn't stop growing completely because unless you target the glutamine, it's going to continue. It can live. It might not grow very much. It grows much slower. So you can take a very angry and inflamed, rapidly growing tumor and lower the blood sugar, and the tumor grows much slower. It becomes less angry and less inflamed. But it doesn't necessarily go away completely. We've had people that have had these tumors in their body for years, always there, but never going away, but not so aggressive or lethal.
I remember I read a paper where you published where the higher the blood sugar, the faster the tumor grows, and the lower the blood sugar, the slower the tumor grows.
Yes, we were the first, I think, to publish this back in 2002, where we showed a direct relationship between how high blood sugar is and how fast the mouse tumor in the brain would grow. So we showed that the lower the sugar, the slower the growth, and the higher the sugar, glucose, of course, the faster the tumor growth. Then we started to see many of similar kinds of studies done in humans with all kinds of different cancers, breast cancer, colon cancer, brain cancer. The higher the sugar, the more rapidly the tumor would grow. And the lower the sugar, the less rapidly the tumor would grow. And it was published in many, many papers showing this relationship between the level of blood sugar and how fast the tumor would grow. And this is why I don't understand how some oncologists or even physicians would say that glucose has nothing to do with the growth of the tumor. It absolutely does. It's in the literature. A big problem, another reason why we're not making any advances, is the people who should know the information were never told of the information, nor do they read the scientific literature. So that's another reason why we have this catastrophe in cancer, is that people don't read the scientific literature, or even worse, don't even understand it. So, and then we have the tragedy.
What blood sugar level did you observe where the tumor doesn't grow?
That goes to the glucose ketone index calculator. And don't forget, the brain is largely responsible for taking in most of the sugar in our body. Okay, the brain. However, the brain can switch over to ketone bodies when you lower blood sugar. So, but the cancer cell can't switch over. So this is the difference. So many of our heart and muscles and things like this, they can all switch to ketones and fatty acids while you lower blood sugar. But the brain, even when you lower blood sugar, the brain will take what's available before any other organ will. The last group of cells that will get the sugar will be the cancer, because if you exercise, muscle takes sugar out of the bloodstream to build glycogen and burns it in the muscle. The brain takes what's available, and the cancer cells are the last one to take what's left. So even if you lower your blood sugar down to a low GK, glucose ketone index is what we call it, you say, well, you didn't get rid of all your glucose. That's true, because the muscle and the brain need it before the tumor cell needs it. So the last cells to get it are the tumor cells. So when you lower down, the brain and the muscles are going to take what little you have in there, and the tumor cells are going to get much, much less, and they're going to die. So exercise and low GKI puts a tremendous stress on the availability of the tumor cell to survive. And then you simply come in with a drug that targets the glutamine, and you slaughter these tumor cells, slaughter them. So, and you keep the body healthy while you're doing this. And gradually the tumor gets smaller and smaller and more indolent, where a surgical procedure or radiation procedure or some final procedure with an immunotherapy could potentially cure the disease, potentially. But you put the metabolic pressure first, and then you come in with the other strategies to remove the final survivors.
And which type of exercise do you find better in lowering cancer? Cardio or strengthening or a combination of both?
Probably a combination of both. And you measure the glucose ketone index while you're doing this as well. So yes, it's a combination of both because you're moving muscles. Muscles take in glucose from the bloodstream. So the more exercise you give, whether it's aerobic or static weights or whatever you want to do, it's all effective.
Mhm. And can you reach this ketogenic status without the ketogenic diet through other diet like fiber-rich diet?
Well, you can get, it's again, what diet do you need to bring your GKI down, the glucose ketone index? Um, as I said, any diet can do that.
Mhm.
If you're a carnivore, you can do it just eating meat because there's no carbohydrates in the meat, very little. But you'd have to then slowly restrict because you have to get where you're going to get your ketones. Yes, fat would certainly bring it in, but your body will start mobilizing stored fat in our adipose sites, and they will go to the liver, the fatty acids, and the liver will make ketone bodies. So if you don't bring carbohydrates into your body, uh, you have to then generate carbohydrate from burning other fuels in the liver, and the liver will produce a small amount of carbohydrate through gluconeogenesis, but the brain will gobble that up if you have low glucose. The brain and the muscles will gobble it. The tumor cell will not be able to get much from that. So, but you can get a low GKI with a Mediterranean diet, a carnivore diet, a vegetarian diet, a fish diet. Doesn't make any difference what you eat. You would just measure your blood ketone index, the glucose ketone index. A ketogenic diet is simply a diet that has a lot more fat than protein or carbs. That's one kind of diet, but a carnivore diet. And then each person is a unique metabolic machine, a metabolic machine. So what a diet that may work good for one person may not be good for another person. That's why we use the GKI as a marker to determine whether you're going to put pressure on your tumor, regardless of what diet you eat. And people say, "Oh, I can't get a GKI low. You said 2.0, I can never do." Well, then go on water fasting for crying out loud. That'll bring it down. It'll bring, and then add stuff back and see, oh, I'm starting to eat this particular food and it's not rising my GKI, then good. That's a kind of a food. All the foods, regardless, must be balanced in macro and micronutrients as well, so you don't cause an imbalance.
And if someone cannot tolerate ketogenic diet and wants to reach this ketogenic state, what can they eat?
It depends on each person. Some people can't tolerate ketogenic diets, but they can tolerate a carnivore diet or a fish diet. It depends on the person. What is the best way to keep the person relatively happy or not stressed out and killing cancer cells? And everybody has to determine that for themselves. They always ask me, "Oh, he never said what to eat." I cannot tell you what to eat because you might be different from the other guy. He said to eat that, and I ate that, it didn't work, and he ate that and it worked. Just use your glucose ketone index with the Keto Mojo meter, and you'll know what you can and cannot eat. And that varies from one person to the next. So as I said, if you have a diet that's balanced appropriately in micro and macro nutrients and low and highly processed carbs, it's probably pretty good for you. Eat a little bit of it and see what it does to your GKI. And then you'll know whether or not you're in the zone of therapeutic ketosis, which is healthy, keeping your mitochondria healthy, reducing oxygen-free radicals, and reducing the risk for cancer and other chronic diseases.
Is there a diet that lowers glutamine?
Diet will not lower glutamine. Exercise can lower glutamine a little bit, but no diet can lower glutamine. Your diet can lower glucose, but a diet cannot lower glutamine. Therefore, it's a press pulse, and the pulsing comes from drugs.
What are some of the agents that lower glutamine, and which one did you find to be the most effective?
Well, you can lower. We actually found that the drug 6-deoxynorlucine, it looks like glutamine. That's in our research. That's the most effective so far. which targets the ability of glutamine to be metabolized to glutamate. So it blocks that first metabolic step. That drug was in fact used for many years on children and people with different cancers, but it was discontinued because they said it was toxic. It's not as toxic as the red devil, doxorubicin, and all these horrible chemicals. Um, it's off patent, so people can't make money on it. But the other thing too is when they did use DON, which is deoxynorlucine, they never targeted glucose. They did, they didn't do it right. They didn't do it right, and they didn't transition the body to ketones. And therefore, the most important drug that we know of to manage cancer is not being used because they didn't do it right. So, but the drug companies are making other drugs that can target glutamine, and these are coming on the market, but they don't target the glucose and put the body in nutritional ketosis when they're using their drugs. So the efficacy of these drugs can be some efficacy for sure, but they're not going to be maximum unless you target the glucose when the body is in nutritional ketosis. How is that so hard to understand? I'll tell you one reason. They want to make money on the drug, and they have to show that the drug by itself has therapeutic benefit. But if I put, so it's more important to make the money than it is to keep the person alive. This is the bottom line. So people have to know that. I'm more interested in seeing how long I can keep people alive because that, to me, tells me we know what we're doing. And it's a use of different drugs, different diets, different procedures that help each person as a unique individual live longer with a stage 4 cancer or any kind of cancer for that matter. So I'm more interested in keeping people alive than making money. I have a job. I make money from my job. I don't need to make $1 billion. All I have to do is keep people alive. What's wrong with that?
Yeah, well, thank you for that. That's why so many people follow you. Do you find the metabolic therapy to be effective in certain types of cancer and to be less effective in other types of cancer?
I think it works with all kinds of cancers equally. Maybe you have to modify it slightly. We just published a big paper for managing glioblastoma, terrible brain tumor,
Yeah.
a very, very comprehensive treatment protocol with myself and Dr. Thomas Duryea as the first author, and many of our colleagues, MDs and PhDs and nutritionists on the same paper. And we went through all of the different ways in which we can reduce the growth of rapidly growing glioblastoma and put it under control. But the reason why the diet targets glucose and glutamine while transitioning the body off to ketosis, since every major cancer that we have looked at has the same problem metabolically, they're dependent on a fermentation and they have defective number, structure, and function of mitochondria. I predict, and where it has been done, in lung cancer, colon cancer, breast cancer, yes, metabolic therapy will be the standard of care for all major cancers. Once the ideological dogma, once we get rid of the dogma, so that cancer is a genetic disease, we'll start to really come after it, all these major cancers, and we will drop the death rate significantly if this were to be started in the way we have defined it and designed the treatment protocol.
And I believe I read an article where you looked under the electron microscope for the majority types of cancer. Could you tell us what were your observations?
Well, I didn't do that, but I went back for those people that did do that. The work on the electron microscopy, you can't see mitochondria very clearly with a light microscope. Even at 400x, 400 times, you can just see specks and things. But in order to see the organelle really close up, you need electron microscopy. So when electron microscopy became available in the 1950s and 60s and 70s, when they constantly perfected this instrument, many pathologists and scientists were simply excited to look at the different structure of organelles under the electron microscope. And they didn't really know the significance of what they were looking at, most of them. Some of them, of course, did. But you found mitochondria that were broken. You found mitochondria that were abnormal in structure. Sometimes you have very, very few mitochondria. Other times you have tremendous numbers, but they're all morphologically abnormal. And in our understanding of biology, structure determines function. So if the structure of the organelle is abnormal, the function of the organelle will be abnormal. And what is the function of the organelle mitochondria? Is to produce energy efficiently so the cell can maintain a homeostatic, differentiated state. So all the tumors that we have looked at, that others have looked at and reported, have all shown abnormalities in the number structure of the mitochondria. And then when you take the mitochondria out of the cell and you begin to study the mitochondria separately, you find it's functionally abnormal as well. So that's telling us that the very organelle responsible for maintaining homeostasis is abnormal, causing the cell to fall back on a fermentation dependent on glucose and glutamine. So that's the linkage. Structure determines function. When the structure of the organelle is abnormal, the function will be abnormal, and that will cause the cell to rely on a fermentation metabolism to grow. Dysregulated cell growth driven by a fermentation metabolism because the organelle that's supposed to maintain differentiation and metabolic homeostasis is damaged, is abnormal.
And could you talk to us, please, about the experiment where they took the nucleus from a cancer cells and put it in a normal cell, and took the nucleus from a normal cell and put it in a cancerous cell?
Nuclear mitochondrial transfer experiments
Yes.
were telling us that if you take the nucleus out of the tumor cell, isolate the nucleus from the cytoplasm of a tumor cell, and put that nucleus into the cytoplasm of a normal cell, and then put that cell under the skin of a rat, you don't get any growth. You don't whatever happened suppressed whatever was in the nucleus that was thought to cause dysregulated growth. It was reduced, was removed. On the other hand, if you take the nucleus of the normal cell and put it into the cytoplasm of a tumor cell, and then put that under the skin of the rat, you got dysregulated growth on a big tumor. So clearly, what it was telling us, it is the mitochondria in the cytoplasm, not the mutations in the nucleus, that are causing the dysregulated cell growth. And this has been done both in vitro in culture dishes, as well as in living animals, frogs and mice, all of these things. And then on the other hand, if you take an aggressive tumor and remove the mitochondria from the cytoplasm and put in new mitochondria from a normal cell, you get a suppression of the dysregulated growth. On the other hand, if you have an indolent tumor growing very, very slow, and you remove the mitochondria and put the mitochondria from the aggressive tumor into the cytoplasm of the indolent tumor now becomes super aggressive. So clearly, dysregulated cell growth is due to abnormalities in mitochondria structure and function, not in the mutations in the nucleus.
That supports that it's a metabolic disease rather than a genetic disease.
That's the strongest evidence that we know of. And then we also know that you have to realize in the genetic dogma that you have many thousands of different kinds of mutations that can be found in tumor cells. And not all the mutations are considered drivers of the dysregulated growth. Only some mutations were thought to be drivers. They call driver because that mutation is more important than the passenger mutation, which just doesn't do anything. So what we're now finding, so the genetic field said, oh, cancer is a genetic disease, but only due to driver mutations. So if you have these drivers, that would lead to dysregulated cell growth. But now new information is showing that normal cells in our body, you, me, we have mutations in driver genes that are not causing any dysregulated cell growth. So that's another piece of evidence that completely disengages the gene theory, that we have driver mutations in our body that don't form cancer. And now we have some cancer cells that have no mutations. So yet they're dysregulated in the cell growth. Not too many, very rare, but it does happen. So clearly what this is telling us, the nuclear mitochondrial transfer experiments, the findings of driver gene mutations in normal cells, All of this is telling us the cancer cannot be driven by mutations. Dysregulated cell growth is not caused by somatic mutations. And the germline mutations that are risk factors, all of the germline mutations disrupt mitochondrial energy function. So clearly, origin of cancer it's a mitochondrial metabolic disease. And the mutations that we see in cancer cells come from the abnormal metabolism of the mitochondria. So reactive oxygen species are carcinogenic and mutagenic, and they are coming out of the abnormal mitochondria, and they cause the nuclear gene mutations and broken chromosomes and this kind of stuff. So clearly, when you put all the parts together, cancer is a mitochondrial metabolic disease driven by a fermentation metabolism involving glucose and glutamine and an inability to transition over to fatty acids and ketone bodies. How do we know that? I did the experiments. I showed the evidence and published the evidence in the scientific literature. And then you say, well, how come nobody wants to? Because they're dogma. The dogma. The brain can't think when it's indoctrinated. And the jobs and the money and the billions and billions of dollars locked into a failed theory. And we have 1,700 people in the United States dying every day because the brain can't get out of the dogmatic view. It's an ideological dogma. And I tell you this, I show you all the evidence. I did the experiments. I show the efficacy. Can't be true. Can't be true. You tell me what's wrong with that picture. You tell me what's wrong with this picture.
I mean, it's difficult to argue against anything you said, and you have explained it very clearly. Do you have this sort of conversation with oncologists, and what is their reaction towards these hypotheses?
They get pissed off. A lot of them get angry. They get angry because they said, I never heard this and never taught this in medical school. Oh, because you haven't heard it in medical school, that means it can't be true. And some of them are very good. They say, well, we got to learn. We have to know what you guys know. And I said, yeah, that's going to drop the cancer death rate. That's going to manage the disease. A lot of reasons for why what I said cannot be accepted. You have a multi-billion-dollar industry running on a somatic mutation theory of the disease, and it's not that. And yet everybody's making a lot of money on this. And then you come along and say it's a completely different disease than what you thought. Oh, how are we going to make these changes? But don't forget, we're not getting rid of standards of care. We're still keeping radiation, chemo, surgical mutilation. Surgery can be done as a final stage for the cure because you can cure cancer with surgery. If you can take out we want to bring the tumor down, make it less angry, small, like a wart, something really small, less angry, less inflamed, and then you can surgically debulk it. So surgery is still part of this. Yes, we can still use radiation. Yes, we can still use chemotherapy. You just have to know the order in which you need to do these things, and that requires a retraining. So, and understanding what you need to shrink your tumor down, make it indolent, weak, and now come in with other therapies to potentially deliver metabolic therapy is not likely to produce a cure, but it can certainly allow the combination of all of the therapies together to lead to a cure. So, but right now we're not doing it. We're not doing it correctly. We're using therapies up front based on a somatic mutation theory that's not effective. You have to rearrange the order in which you attack this tumor in the right way. You want to make sure there is no toxicity in the treated patient. They just don't know how to do what we already know how to do. And once our word gets out, you're going to see incredible responses on these patients. So is it going to be quick? No, it's not going to be quick. It could take you 6 months to a year to manage cancer in the correct way.
Yes.
But when you emerge from the treatment, you'll be much healthier than you were when you started. Not only will your cancer be under control, but your whole body will be healthier.
And for those who wants to lower their risk of cancer without advanced tracking, they don't want to get pricked multiple times
Well,
a
it
day.
wouldn't have to be multiple times a day. We're working that out. Yes, they have technology is improving. We already have the free Libra for measuring blood sugar, and you can measure it. It sticks to your arm, and it measures glucose in real time, and then it goes to your cell phone. So you can actually see your blood sugar at any given time. Eat something, eat a banana, watch it go through the roof. Eat a small piece of butter and watch it go down. But we don't have the ketone yet. the monitor for ketone. You still have to prick your finger. That's why the glucose ketone index, the Keto Mojo, and maybe some other meters can measure the two together. And I know people don't like to prick their finger to, I don't like it either. But, people who have diabetes, they have to do this many times a day sometimes. But now they have the Libra. And I think once we have a free Libra ketone meter, then you can get real-time GKI on your phone, and that will tell you. Then you'll have right there on your cell phone. You can actually eat something and watch it change on the cell phone. Then you'll know what you can and cannot eat.
And you believe this might come out soon? GCM type of machine that detects ketone on top of blood sugar?
Well, if people want to make a lot of money on the machine, they certainly would be driven to make it fast. That's not my realm. I don't know how to do that. I just build the structure of what needs to be done. But technology improvements will have to work out these meters. And those guys are going to make $1 billion once they get the free Libra ketone together on the blood. Because you can not only manage diabetes, you can manage cancer, dementia. You can put yourself in a completely new state of health doing this. But I don't know. Yes, there are people out there, believe me, there are people out there that want to develop this as quickly. We developed that, but we couldn't make any money on it. It's unbelievable. You can't patent a formula. Can you believe this? You can't patent a formula, but you can take that formula and put it into a machine that will do it, and you can patent the machine. But I didn't do any of that. So again, our goal was to keep people alive. Well, I mean, that was our goal. Let our industrial technologies work and build all those kinds of things.
And when you're trying to stop the cancer cells, is the aim to get rid of it completely, slow it down, shrink it?
Think that, well, the goal, of course, is to eliminate it completely. We are not there yet. I can't be sure. We have a number of people who have used metabolic therapy and are declared no evidence of cancer. We had one dog actually that was cured of mast cell tumor. I published that paper. The dog, you only know when you're cured is if you die from old age and cancer doesn't come back. You got a tumor, you use metabolic therapy, and it goes away, but you're always worried that it might come back, and you live to be 98 years old and you die from old age and the cancer never come back, then your children will tell you were cured from the cancer because it never came back. But it's interesting. With metabolic therapy, if known, you can use it to prevent cancer. Of course, you don't need to target glutamine to prevent cancer. You just need to keep a lower GKI and switch from high carb to a fat, a ketone. So you can keep your mitochondria healthy as a prevention. You can do this as a prevention. And then if you were to get cancer, of course, you can come in with metabolic therapy now, targeting the glutamine and making it a little bit more effective in the strategy. But it's still a metabolic approach. And so people can use metabolic therapy to both prevent and treat cancer.
And what is your opinion about intermittent fasting? And do you do any intermittent fasting yourself?
Oh, yeah, I do it. Yeah, oh, sure. I do a lot of exercise and intermittent
do you do
fasting.
12 to 16 hours, or what type of fasting do you do?
I do 18, 20 hours. So you eat at night and then you don't eat again until the afternoon. You go out and do a lot of exercise. So I'm trying to do what I can to keep the beast away. But if I do get cancer, yes, I will have to embrace my own theory, my own therapy. And what we do is we'd have to go on a low carbohydrate diet for 12, 14 days, get my GKI down, and then I will take Dawn. And my friends will deliver it to my physician friends will deliver it to me, and I will do exercise, and I will keep my body as healthy as I possibly can, and hang on, because you're killing these tumor cells, and you go down to the blood, you get your blood work, make sure your blood work, all your blood, and you should never be out of blood homeostasis. All the markers should be in the zone. I keep a low GKI, water only fasting together with zero carbohydrate diet, and then pulse the damn glutamine targeting, and then hang on, baby, hang on, and see what happens. Um, and now, of course, we still were quite healthy, eating natural grains through the natural carbohydrates, complex carbohydrates, and the exercise that we got. But now we are saturated with highly processed carbohydrates, which are refined sugars and these kinds of things. And we're less and less active in our daily lives, and we have more and more carcinogenic compounds, forever chemicals, microplastics, toxins in the air. So we're barraged. We have a very poor diet and lifestyle, and we're barraged by chemicals from the environment, which I think is contributing to the earlier and earlier onsets of cancer, together with the diabetes, the obesity epidemic, the obesity which is striking all major societies now. Um, and this puts us at risk for all kinds of chronic diseases, cancer being the number one chronic disease, which can be also significantly reduced. You can reduce all these risk factors for cancer and diabetes by bringing your body back into a state of nutritional ketosis, intermittent fasting, exercise, all these kinds of things. And I know people don't want to do that. They don't want to do that. They enjoy eating but I think there's nothing wrong with natural carbohydrate foods, natural sugar rather than highly processed foods. Everybody likes a pastry every now and then, a pizza, whatever it is. But you have to measure your GKI while you're eating the pizza. You might not be so enthusiastic to have another slice.
Even these animals, they love the pizzas and donuts, right? You have talked about it before in the San Diego Zoo.
Yeah, the zoo in San Diego. So not only the zoo in San Diego, also the zoo here, the Franklin Park Zoo here in Boston. I did both. I talked to the vets in both of them. The bonobos, they have these pygmy chimpanzees and orangutans, but once a week they take a little honey and they put it on a small dish, and they hide the dish in the different parts of the cave. As soon as the temperature and the bonobos hear the guy out there, and they know kind of what time of the week they're going to put out the dish with the honey on it. Little dish. They come flying out of the cave, running around, looking under the bushes and the rocks, looking for that one little dish that has the honey on it, and they start licking it off the dish. So I said, why don't you go down to the donut shop, the Dunkin' Donuts, get a box of jelly fills and crullers and all, throw that into the bag. They'd be out there eating those things so fast. But the veterinarian said it would be animal cruelty. You'll be
Yeah,
damaging their health.
damaging the animals' health, but not for us.
You put the damn bonobos at risk for chronic disease by giving them donuts that humans are eating every day.
Yeah. There is a neuroscientist who did a study in Africa where these baboons or monkeys were eating leftovers from human. So they were eating highly processed carbohydrates, chips and biscuits. And interestingly, a lot of these animals started to develop type 2 diabetes and hyperlipidemia, metabolic syndrome and obesity and all kind of metabolic diseases.
Now you want to tell we can't do that experiment because it's animal cruelty. Again, animal cruelty. But yet we're doing all this stuff to ourselves and all smiling about it. And then we get all these chronic diseases. But human cruelty.
Yes, yes. And isn't it insane that these animals start to develop metabolic syndrome and type 2 diabetes and
Yes,
hyperlipidemia?
absolutely, absolutely. But we're not allowed to do those experiments on control groups of monkeys, because monkeys are sacred animals of some sort, and we do the experiment on ourselves. We already know what's going to happen. Type 2 diabetes, metabolic syndrome, cancer, macular degeneration, cardiovascular disease, dementia. We're going to get all those things. We already know what's causing it, but we just choose not to stop the cause.
And there are these cases where someone is healthy, exercise, fit, eats healthy, and then develops cancer.
But when people say I'm healthy, I exercise, we don't really know. Did they measure their GKI every day? Was their GKI? I don't know what they say. Oh, I'm a healthy guy. I run, do this. What's your GKI? I don't know. Well, how do you know you're really healthy? How do you know you're in a nutritional ketotic state? because I said nutritional ketosis is what keeps the mitochondria healthy, because that's the way we evolved as a species. We always were in a state of nutritional ketosis. A
Mhm.
guy who exercises, I don't know what he's eating. What his GKI is. Show me a guy who's maintained a very low GKI, eating no highly processed foods, does a lot of exercise, and then develops cancer. We want to know about that guy. But people say, "Oh, I'm healthy." Who's tell you you're healthy? You tell me you're healthy. You don't. I don't know. I didn't look at your physiology. Oh, my blood work looks good, and maybe he is. But what's your GKI? Oh, it's about 50 or 40. No, no, no. You're not in the healthy zone. So, but anyway, we need to know that we don't. And the answer is we don't have any scientific evidence to prove that a guy who looks healthy is actually in a state of nutritional ketosis when he developed the cancer.
Mhm.
And we need to do that. We need to look at all these things more.
Mhm.
And that also, generally, those guys that appear to be very healthy generally have lower risk in general than the guy that's morbidly obese. So we know that there's a pretty strong relationship between that.
And you have talked about microplastic and environmental toxins. Is there a way to avoid these harmful substances?
It's going to be hard. I mean, everybody, I'm even drinking a bottle of water here in plastic, right? How do I know how many microplastics are in there?
Yeah.
But they seem to be permeated through our entire food chain.
Yeah.
Plastics, and they were all of us, no matter what country we live in, we're going to be exposed to this. And then there's these forever chemicals that are out there. These are chemicals that our body can't break down, and they collect in tissues. So you got microplastics, you got forever chemicals, you got a poor diet and lifestyle, creating inflammation, making the microplastics or the chemical carcinogen potentially worse. You put all that together, mitochondrial dysfunction, transition from oxidative phosphorylation to substrate level phosphorylation, driven by glucose and glutamine, dysregulated cell growth, and cancer. There are certain environments where we have a lot of contamination in the atmosphere and in the soil and in all the other things that we put ourselves into. And these all of these things impact negatively on our physiology, leading to mitochondrial dysfunction and the whole slew of chronic diseases as the result of that.
Even in the animals themselves, they're injecting them with hormones and antibiotic and the GMO soil and pesticides in the vegetables and all of the environmental toxins. These things has to be cancer causing to a very high
Yes,
extent, right?
absolutely. Now, if you have what we call free range cow, where they eat just grass, and then you find out, oh, it's gamey. Did you ever eat a free range cow, buffaloes, or any of these kinds of things? The color of the meat is actually different. So you have a corn fed cow, the meat is tender, it's juicy. I didn't realize this, but when cows eat cow did not evolve to eat corn. The
Yeah.
cow, the buffalo, they eat grass. They eat grass, they don't eat corn. So they eat corn, they get massive acid indigestion, and they have to keep regurgitating the corn. And then you give the cow a lot of antacids, and the cow eats the antacids to neutralize the acidity in its stomach, and it makes the meat very, very tender. But the free range buffalo or cow, the color of the meat is a darker red, because it's more natural. Now, if they're eating natural grasses that have no herbicides in there, and alfalfa and all these natural grains, then your meat is going to be very free range. It's going to be more organic than the cow that's being driven by hormones and all the corn and corn-fed beef. Oh, people say I gotta have corn-fed beef. Corn-fed beef. It's so juicy. It's so great, and it is. I have to be honest with you, and it's more tender and more juicy than the same piece of cut of meat on a free range cow. So free range is more organic, more less contaminated than the corn-fed or the hormonally produced cow. You're 100% right. All of this is in our food chain, and we taste it. We think one is better than the other, but generally one may not be as healthy as the other.
And the majority of these harmful things are unfortunately unavoidable.
Unfortunately, a lot of it is unavoidable because we can't. We just don't have access. We have convenience, convenience, convenience.
Mm.
Everything is convenience. So nobody wants to walk far to get something. They want to drive there. In our country, you drive up to the hamburger shop or the donut shop, and you don't even have to unask the car. You just sit there, and they pass it through the window. Think of our Paleolithic ancestors when they're having to go out and kill some buffalo all day long, beating the hell out of this thing, chopping it up. And then some guy to go, you can drive into it and they hand you the beef right through the window. You don't have to do anything.
Here we have delivery also.
That's right. They deliver it, run to the
Here
door.
with the phone, with a click.
Yes. DoorDash. You just
Yeah.
call up on the phone, and all of a sudden the guy comes to your house with all this food. You wonder why you have cancer and diabetes and dementia. I mean, all the things that we're suffering with are the result of our technology. And I've told people this over and over again. Our technology has evolved so much faster than our biology. We are still Paleolithic. It takes millions of years to change your biology, but our technology has rapidly evolved.
Are you passionate about the groundbreaking and heroic research of Dr. Thomas Seyfried on metabolic therapy and cancer? So are we. That's why we've created something special for you, in collaboration with Johnny Rockermeyer, a German book publisher and translator. Introducing our collection of meticulously crafted books that distill the essence of Dr. Seyfried's work. Dive into the science and discoveries. These summary books are your gateway to understanding the intricate world of metabolic ketogenic therapy in a clear, concise, and engaging way. Whether you're new to the subject or a seasoned enthusiast, our books offer insights that can change your life. Ready to explore this transformative knowledge? Visit our website at www.cancerasametabolicdisease.com to get your copy. You can buy the ebook there directly and the paper book via the provided links. Here's the best part. A portion of every purchase goes directly to support Dr. Thomas Seyfried's groundbreaking research. That's why the direct ebook purchase is the best option to donate as much as possible. You can see all of the donations Mr. Rockermeyer has already made at www.ketoforcancer.net. That's right, when you buy our books, you're not just
to the point now where we're out of sync. We're out of sync with the natural world. And DoorDash, that's the greatest thing.
And for people who are living a traditional lifestyle, raising their own chicken, growing their own vegetables, this has to be healthier, right?
The term in the dictionary that came out a few years ago, Oxford Dictionary, called locavore. Locavore is a term to describe foods that are grown naturally in the local environment. So people would have a garden or a community garden that would be fertilized by manure, and all the soil would be natural, and the vegetables that would be grown in that, or free-range chickens or animals that would be in the local environment. It's called locavore, which is essentially the way we evolved. We were all locavores because we would kill and eat things in our local environment, which is much, you have to be out every day picking something up from the ground, natural, and bring it back into the house. People sitting in traffic and they're all freaked out. They come in the house. They don't want to go out in the backyard and start pulling stuff out of the ground. They want to just throw something into the microwave. But the locavore is that what you just asked, how do you prevent that? People eating food from the local environment, grown naturally in your local environment. That certainly will give you the nutrition that will keep your mitochondria as healthy as possible. And of course, the foods to try to reduce are the highly processed carbohydrates, which are all in packages. I even have a Twinkie. Processed carbohydrates, baby. Has almost completely, nothing here is natural or healthy. Yet people, not only that, they deep fry these things and then put powdered sugar on top of them. Can you believe this?
Yeah.
And they smile while they eat them.
And if the mitochondria gets damaged, is there a way to reverse it? Is it irreversible? Are you trying just to preserve the functionality?
We and Warburg both said there is a threshold upon which, before there's a certain destruction in the mitochondria, it can be reversed up until this threshold point, which we're not really sure about, in which time it becomes irreversible. I am not in the business of trying to reverse cancerous mitochondria. I kill them. I'm not a friend of the tumor cell. I'm the worst enemy that the tumor cell can have. My job is to kill the tumor cell. However, if we can protect the normal cell from ever becoming damaged mitochondria, then we can prevent that. But once you have irreversible damage to the mitochondria, I'm not in the business of re-educating a mitochondria, because it can turn back. It can recidivism. It can turn back on you at any time. I'm not interested in that. We get rid of them, kill them, get rid of them from the body. That's my view. That's my view of managing cancer. We prevent cancer, but if we get cancer, we kill them. And we kill them by starving their fuels that drive their dysregulated cell growth, which is glucose and glutamine, while transitioning the body over to nutritional ketosis, and then come at them that way, and eventually purge.
From your observation, when you target these cancer cells, do they disappear, shrink, slow down? What happens with these cancer cells?
We did this. So all three of what you said, they shrink, they die in different. You can see the dead cancer. We did that. You can look at dead cells under the microscope and see the dead cells. When we did glucose glutamine targeting with Don, we saw massive numbers of dead cells, but we dead cancer cells, of course, in our preclinical system. And we saw some cells in other cases in humans where a lot of them were killed, but they were not all dead, and that's where you have to move the process forward to see if we can kill the rest of them. But here's the situation: if you kill a lot of cancer cells all at the same time, you have all these dead cancer cell bodies laying around in a particular tissue. Our immune system is designed to come in and gobble up and remove dead cells. That's part of what our macrophages do. Macrophage is a type of immune cell that will be effective in wound healing. So we kill a lot of cancer cells. The problem is if we are too aggressive on glutamine targeting, we prevent our immune cells from picking up the dead bodies, the dead cancer cells. That's why it's absolutely crucial that when you target glucose and glutamine and kill a lot of cancer cells, you have to pull the glutamine targeting off and let our immune cells get back to their normal job and picking up the dead bodies. If you're too aggressive on the glutamine, you kill a lot of cancer cells, but prevent the immune system from cleaning up the microenvironment, therefore leading to an infection and making another kind of a problem that you shouldn't have in the first place. Again, we have such a broad knowledge of cancer. We are able to track down those cells and kill them without harming the rest of the body when we know what to do and how to do it. Because I've looked at all these things. I have answers to your questions, mainly because I did the experiments next door with my colleagues. And we exactly do what you asked. What happens to these cells? Where do they go? What's going on with these things? Yes, we looked at them. And we saw exactly, some of them are shrunken. Some of them are outright killed. Some of them just hang on. We didn't kill them all. So that's why we're still perfecting the procedure.
Well, thank you for that. Could you share with us the story of Pablo?
Pablo was a good person. He passed away just recently, last August, from his third debulking surgery. He came to me in 2014 with a glioblastoma, and he was a young man. I think he was only 24 years old. He had diagnosed with a glioblastoma, and he didn't want chemo radiation. He didn't want any kind of a therapy. At that time, we were really very, very new in what we understood. In fact, we didn't even know completely about targeting glutamine at that time. We knew it was necessary, but we didn't really know how to do it too much in human. But he just went on a really low GKI. He got his glucose ketone index down. He ate avocados and eggs and no carbohydrates. And his tumor shrunk. It became indolent. And they said, well, if he didn't have radiation and chemo, he was going to be dead in 12 months. He didn't want radiation chemo, so he said, I'll try the metabolic approach. And he said they told him it was an inoperable tumor, that he had to have radiation and chemo to control it. And he said, well, you can't cut it out. I don't want radiation. I don't chemo. So what happened is he went on a metabolic therapy, and then after 2.5 years of just metabolic, the tumor became less angry. It started to grow slow, but when it grew, it became more circumscribed. And the neurosurgeon said, I think we can get this tumor out. So he went for a surgery in 2017. They took the tumor out, and then they said they think they got it all. So he goes back and starts eating carbohydrates again. And then all of a sudden, what was thought to be not there, something starts growing again. And then he immediately went back on metabolic therapy. And then the tumor continued to grow. He never targeted glutamine. He just targeted the glucose. The tumor continued to grow again for another 3 years. And in 2020, he had another debulking surgery for a tumor that was considered inoperable. And then it continued to grow again. And then in 2024, which was last year, he had another debulking surgery, and then we spoke to him in August uh, after his third debulking surgery, and we were laughing. He was very, very healthy. We were joking around. I said, Pablo, you were supposed to be dead 10 years ago. 9 years ago, you were supposed to be dead. He got married. He had two children. Um, and we were laughing hysterically because he was diagnosed with an inoperable tumor that had now been operated on three times. So then what happened? We didn't know, to our knowledge. I said, he could outlive me. I said, that, you keep living so long. I said, you're supposed to be dead. You're a guy with a glioblastoma living 10 years,
That's what
10 years.
that's what they told him,
And
right? In the
huh?
beginning, 9 months That's what they told him in the beginning when he went to.
Yeah, but then I guess the neurosurgeon said, we still have a tiny little piece of tumor left, and we think we can get that out. And he didn't tell me or my colleague, Dr. Duryea. And and then we got an email from his wife saying that Pablo just had another surgery. And I said, wow, I didn't know that. And then we have a picture of him coming out of the ice. He has both thumbs up. He's smiling. He talked to his wife. He was chatty. He was really great. And that night, he had a cerebral hemorrhage from the surgery.
So he died from a surgical complication rather than the
Yeah,
cancer.
the surgical problem rather than the cancer. So I don't know how long, if he didn't have that last bit of surgery, I don't know how long he could have survived. He certainly wasn't dying from the cancer, that's for sure.
Did he receive any glutamine lowering therapy?
No, he did not. He was against all kinds of drugs. So he just wanted to do what was working. And I told him, I said, you got to get some embed. We know now these parasite medications can target glutamine with much less toxicity. They're not as strong as Don, but they do also work. And he didn't take anything other than the metabolic therapy. So I think we can do much, much better on other cancer patients if we do what we know how to do. I think we'll even get much better results. But the fact that you got a guy with a GBM, he also had an IDH1 mutation, which produces a metabolite called 2-hydroxyglutarate, which actually targets glycolysis and shuts it down. So he had a therapeutic gene mutation, which I think the field needs to spend a lot more time on. They're always trying to target this. And even some of these fools try to target IDH1, which is actually a therapeutic I don't want to go down that path because you have no idea how little knowledge people have of the biology of cancer, and they just do stupid things when they shouldn't be doing those kinds of things.
And surgery can be effective in curing cancer, especially if the cancer is small and localized, right?
The more you can cut out and the more successful you are at debulking, that is removing the tumor, the longer you're going to live. You can cure cancer with surgery. There's no question about it. If you are able to cut the whole tumor out, no matter where that tumor is, and you get every single cell, and it's all circumscribed in one area, you cure the person. There's no question about it. It's the metastatic, it's the spreading of the tumor cells into the local surrounding environment or into other organs that makes it a much more difficult disease. And we discovered that that cell is the macrophage itself. It's a member of our own immune system, corrupted to use glucose and glutamine in a dysregulated way. So I know this. I know how to kill metastatic cancer cells. Our metabolic therapy will do it quite effectively. But when you think cancer is a genetic disease and you're looking at all this crazy stuff, you're not going to make you're not going to drop the death rate any significant way. Yeah, every now and then you get somebody who responds really well, but the majority of people don't respond. They end up dead, one way or the other. Either they're killed by the therapy or they're killed by the tumor. Either way, the probability of long-term survival after a stage 4 metastatic diagnosis is not good. And we think we can keep these people alive a hell of a lot longer because we understand the biology of the problem. We understand this. I busted my ass for decades looking at this stuff. I'm a biologist, geneticist, biochemist. I know we look at this stuff and I talk about it with leaders on the field. My friend Christos Chernopoulos from Semmelweis University, world expert on mitochondrial substrate level phosphorylation. You got to work with the people that know stuff. When you're working with people that know stuff, you can make major advances in science. When you're working with people who don't know anything and have misinformed information, you're not going to make major advances. Just that simple. It's just that simple.
Is there one study where the evidence was so overwhelming, if there is an oncologist or a physician listening to us, that there is one study that you can reference them to?
You have to look at all the studies together, and you have to be able to connect the dots. And that's what I did. Okay, I spent a lot of time looking at Warburg's theory of cancer. I proved that he was correct, and I also showed where he went wrong with our colleagues where he made mistakes, and he didn't know that there was a second form of fermentation inside the mitochondria itself. So now we have cleared up a lot of the mystery of what cancer is based on Otto Warburg's seminal work. I also, as a geneticist, knew right away, pretty clear, once I started doing the research myself and then looking at the nuclear transfer experiments and all this other stuff, I knew right away this can't be a genetic disease. So once you eliminate one of the big stumbling obstacles in the way, then you can really start focusing on what the real issues are. And then we realize we can put all the dots together and we develop a therapy that we published that will eventually become the standard of care. How long will that take? I don't know. We already published the paper. We already set out the protocol, what to do and how to do it. And there it sits in the scientific literature right there. So you want to make it, read that paper, have the oncologists and physicians' assistants and the whole operation follow what we are saying. You will drop death rates within the next several years. Here we did. We published. What more do we want it? What more can I do? What more can we do? We already showed what's going on. We have some big papers. One of our papers just came out the other day. It's already got 16,000 views for the first time, showing that cancer cells are getting energy inside the mitochondria from a fermentation mechanism. The field did not know about this. That's why the glutamine has to be targeted. We have an outline. We have a clear outline supported by hard scientific evidence that we can drop the needle, move the needle significantly in dropping the death rate of cancer. The problem is that these are not, the interference with what I'm saying has nothing to do with science. It has to do with policy and acceptance of the scientific facts, not the science itself. The science, to my mind, is quite solid, and it's based on decades of research.
Yeah. And are there ongoing clinical trials at the moment?
No, there are no clinical trials because I told you the hospitals think cancer is a genetic disease and they haven't figured out how to make a profit on that. Right now, the medical institution is not in the position where they want to change the standard of care. It's very, very profitable. People making a lot of money, hospitals, pharmaceutical companies, oncologists, they make a lot of money treating cancer. And now you come in and say, what are you going to do? Water-only fasting and use repurposed drugs? Who's going to pay for this? You know, the only people to benefit from metabolic are the patients. The system is not yet ready to accept a therapeutic strategy that's effective and not toxic and cheap. You know, bottom line, it's a massive, expensive industry, and everybody is doing really well financially, except the patients. So, you know, you're not going to change until there has to be an outcry on the part of the world population to say, we want metabolic therapy. The problem is that the field doesn't, they're locked into an ideological dogma thinking it's a genetic disease. And therefore, the field all are trying to target genes. And then you have 1,700 people a day dying as the result of a misunderstanding of what the nature of the disease is. It's not complicated. When is it going to overtake? It should never, ever overtake heart disease, ever. We can drop those death rates by 30% in 5 years, 50% in 10 years. Once you start learning how to do metabolic therapy and running the clinics that should be the way it should be done. Now, if people don't want to do that, so it has to go from policy. It has to go. Yeah, I just heard the other day, I was saying, oh, we're going to make vaccines and we're going to do all this kind of crazy
stuff now they're talking about.
Yeah, we had Big Watson computer one time. He could beat anybody in chess. And he win all these games. So they brought him in to figure out cancers, the gene mutations that might be causing cancer. And he was reaming, smoke coming out of the machine and doing all this kind of stuff. And then he made recommendations to the oncologists. The oncologists were smart enough to know the recommendation from this super intelligent computer would have killed cancer patients. So no one told the machine that cancer is not a genetic disease. The goddamn machine was too stupid to know that. So AI is only as smart as the guys that are trying to work with it. So and then if you go on the web and say, ask AI, is cancer a genetic disease? Yes, cancer is a genetic disease. Then you go on to AI, is cancer a metabolic disease? Yes, cancer is a metabolic disease. Well, it can't be both at the same time. It's either one or the other. Is the Earth the center of the solar system, or is it the sun the center of the solar system? You ask AI, AI would say, oh no, the Earth is the center of the solar system and the sun revolves around the Earth. And the AI, what about the sun? Oh, the sun is the center, and all the planets revolve around the sun. So I mean, again, it comes down to the theory. If the treatments are based on an incorrect theory, the outcome is not likely to be effective. And that's what we see, as we said. And right now, the pharmaceutical companies are gearing up that in 2050, 1 out of 2 people are going to have cancer. More and more young people are going to have cancer. The profits are going to be enormous in cancer. We can drop that. We can prevent all that stuff from happening right now, today. We have to do re-educate the oncology community to know that cancer is a metabolic disease driven by glucose and glutamine, and they can't transition over to ketones, so therefore transition the body to ketosis, target simultaneously glucose and glutamine, and bring in your standard of care procedures when you need them.
Yeah.
And that's the solution to the cancer problem,
And
or
there is,
a rational solution based on hard science.
and could you talk to us a little bit about hyperbaric oxygen therapy and cancer?
D'Agostino and I and others, you can't, if you bring your glucose ketone index down, you put a lot of pressure on the tumor cells, they'll produce ROS, reactive oxygen species, from the high pressure oxygen. And then when they're in the low glucose and you give them the glutamine while they're in hyperbaric oxygen, it's a facilitator to kill cancer cells. It's a replacement of radiation and chemo in some ways. And it can make radiation and chemo even significantly more powerful by increasing reactive oxygen species, which will kill the cancer cell.
And how often is the glutamine therapy given? Is there like a duration and frequency?
That's the cutting edge right now. It's called dosage, timing, and scheduling. We are still working on that. And that's interesting because all of our funding for doing this is philanthropy and private foundations. Because figuring out dosage, timing, and scheduling is what they call, oh, this is not sexy science. This is not cutting edge sexy science, but it'll keep damn people alive longer. That's what it will do. But that has to be determined for humans. How long can we hit them with the glutamine targeting, at what drug and what scheduling? And that has to be trial and error with each patient. And then once we see how patients respond, then we can implement it in a more outlined way based on our experience. But we don't know gender. You have issues of age and gender from people, and these all have to be worked out with dosage, timing, and scheduling. We just know what to do and generally how to do it, but the actual specific it's the glutamine predominantly, because we don't have to worry about glucose. But it's that and what better drugs do we? How many different drugs will do the same thing? Targeting glutamines effectively without causing toxicity. What dosage? When do we schedule this? How is the timing? When the patient reaches a certain point, then we come in and do this. This has to still be worked out. And we do it preclinically first in the models that we developed. And then we get some idea. And then I translate it immediately over to the clinic, my physician friends, and they will say, oh, we found out if you do it this way and that way, it'll better come back into the preclinical system and perfect. Feed forward, feedback, feed forward, feedback until we perfect the system. You're not going to have everything right done. We have all the concepts down. We just don't have the actual the precision metabolism for each person.
Well, Professor Seyfried, thank you very much for this incredible interview. I have certainly learned a lot. And thank you for all of your incredible work and research and everything you do. And I want to quote something you have said in a prior interview, and I'm just going to read it. You said, when the science comes, you cannot suppress the truth, and I'm documenting the evidence. And this is exactly what you're doing. So thank you very much once again. And I hope all of your hard work lead to the results and outcome that you are hoping for.
Certainly hope so. And thank you very much for asking really important questions that can help people understand what the issues are. And eventually, you're right, things will change for the better. And that's the hope that we have.